The overall goal of this experiment is to exhaustively characterize MyoD protein partners.
This method can help answer key questions in the skeletal muscle biology field, such as molecular regulation of skeletal muscle terminal differentiation by the key muscle transcription factor MyoD. The main advantage of this technique is that it allows us to unravel MyoD partners that have relatively low abundance when localized in one specific subnuclear compartment.
Though this method can provide insight into regulation of the muscle differentiation, it can also be applied to other systems, such as any other tissue-specific lineage differentiation.
Demonstrating the procedure will be Dr. Lauriane Fritsch, a research assistant from our laboratory.
In this protocol, MyoD complexes will be purified from HeLa S3 cells that stably express MyoD with flag glutenin tags. HeLa S3 cells transduced with the empty vector will be used as the control. Both cell lines were previously grown, collected, and stored at -80 degrees Celsius, as described in the protocol text. Quickly thaw the cell pellets in a water bath at 37 degrees Celsius. All buffers and homogenizers should be pre-chilled, and this entire procedure should be performed in a cold room. Typically, 20 grams of cells are used for each experimental point, but for the purposes of this demonstration, only 10 grams, or about 10 milliliters of cells, will be processed.